going to hand over to Sarah Summer, who is going to be doing the details of the introduction.
Thanks, Tilly. Welcome, everybody, on behalf of the Royal ENTSOC. My name is Sarah Sumner. I am just standing in for our president, Jane Stout, who sadly can't be here today. I'm an RES trustee and the vice president, but I think they only chose me for vice because I'm in London and I can step in and do these things for them. But it's a great pleasure to be introducing our fantastic speaker today, Professor Iain Couzin from the Max Planck Institute of Animal Behaviour in Germany. And in just a few minutes, he will be sharing his insights into the extraordinary dynamics of insect swarms. But before we go on to that, before we let Iain loose on the platform here, I'm just going to give you a few updates about the Royal Entomological Society. We're a growing community, which is absolutely fabulous. We are thriving. We've increased our membership by over 40% in the last few years, which is astonishing. And we're global. We are representing 80 countries around the world. We support our members with a range of events, both online and in person, and through our special interest groups, covering everything from taxonomic groups to broader conceptual topics. In 2024 alone, we awarded £75,000 in grants, and this included research grants, outreach grants, and career development grants. We're very proud of our publication programme. We have 7 journals and an expanding portfolio of books and our world-renowned library, which are all accessible to members. And we also publish Antenna, the membership magazine that gets sent to all members, where we share news and insights from across the insect science community. Our outreach work continues through our work at Insect Week every year. So in 2025, we have that and science fairs, and also the Young Verrall Lecture, which took place last weekend in the Staffordshire Invertebrate Fair, which was delivered by Dr. Ben Clooney. We're also really excited to be expanding into the science policy space with our new Science Policy and Society Committee. And we're also investing in education, including the development of the Royal ENTSOC Garden, which I do recommend you all go and visit in Stratford in East London. So, as I said, we have 7 journals. We're very proud of them. They support a broad spectrum of the ecological community. We have why should you publish there or submit there? We have very fast turnaround times, high quality peer review, open access options available, a global reach, excellent citations, and any profits that are made are fed back into the society. So members will always benefit from that. We have a student forum every year, and I have to say, I've been to a few of these, and they are the best, funnest conferences I've ever been to. So the next one is taking place very soon at the University of Reading. You can register by Friday to participate. It's also available online. And it's an opportunity for students to network, to share their research. It's a really friendly, safe space. There's talks from invited scientists as well as the students. Interactive workshops, lab tours, lots of socialising. The Ento Olympics is brilliant. I'm rubbish at that, but there are people in this room who have phenomenal knowledge. And also, all students get a free membership for the first year, which I think is a fantastic opportunity to step into the society and see what it has to offer. I'd like to highlight Ento 25, which is our flagship conference, which happens every year. It's taking place on the 9th to the 11th of September in the University of Strathclyde and Glasgow, and also online. So please do come along, register now. You can submit your abstracts. They've just opened the call for abstracts. You can also attend online. And it's a fantastic place to meet a huge diversity of people working in all sectors of science, but under the shared umbrella of a love of insects. It's also worth highlighting to you that every year we ask for nominations for the honorary fellowships. So if you would like to nominate someone, an outstanding entomologist who you think deserves this recognition, then please do so by the 30th of May. and our newly elected honorary fellows will be announced in ENTO later on in the year. So before I hand over to Iain, just a reminder that we would really like your feedback on these events. These events are for you, the audience. So we have a lovely QR code. So this is the only time in the lecture when you're allowed to get your phones out. Please take, no one is, that's really weird. Okay, don't get your phones out. We will, if you registered for the event, you will get this link via email after the event. So please do tell us what you think and how we can improve it to make sure it meets your needs. Okay, so it remains for me to introduce Iain. Iain is truly a fabulous person. We were PhD students at the same time, but he's gone on to amazingly great things as director of the Max Planck Institute of Animal Behaviour. He did his BSc in St Andrews, his PhD in Bath with Nigel Franks on ants, did a postdoc in Leeds, and then he was a Royal Society Fellow at the University of Oxford, after which he took up a faculty position at Princeton, and as if it couldn't get much better, he then has had 2 director roles at the Max Planck Institute in Germany. He's got a long list of prestigious awards, and I have far too many to fit on my tiny scrap of paper, but just highlighting a couple. He's been awarded the ZSL Scientific Medal, the National Geographic Emerging Explorer Award, and I'm going to say this wrong, the Leibniz Work Prize, which is a very prestigious prize for German scientists. And so today we are so privileged to have him here delivering us the Verrall Lecture. Iain, over to you.
Thanks, everyone. So we couldn't get the microphone to work, so I'm just going to speak loudly. Is that all right? Is it okay? Good. I'll just turn the lights down a wee bit. Yeah, well, what a pleasure to be here today. You know, as a wee kid growing up in the northeast of Scotland, fascinated by insects, I never ever thought I would be in front of such an esteemed audience. So yeah, I'm going to give a very broad talk today about 2 different systems, cooperative, collective, and a very selfish collective, focusing on some work I've been doing since my PhD. And you'll hear more about these animals. These are desert locusts later on. So collective behavior, which is what I'm really fascinated by, it's really interesting to me because these invisible social connections, the behaviors among individuals, couple the brains. So in crowds, for example, unbeknownst to us, we're strongly influencing each other in subconscious ways. In this particular study, we could see that the gaze of individuals could actually propagate within crowds. It could spread by a process of social contagion. And despite the fact that we are definitely considered fairly smart, at least most of us, there's some very visible exceptions in the world right now. If you look at the gentleman in this elevator, he's unaware that the other people in the elevator are actors. They're in on the act here. And just have a look at how he mimics the behavior of those around him. Don't be smug. We're all subject to this type of social contagion. And so, yes, in many circumstances, we can be very smart. But in other situations, such as in heavily crowded environments like here in Japan, in actual fact, what we need to consider are relatively local interactions among the individuals that give rise to emergent properties at higher levels of organization. So if you look closely at these people in crowds, you may have spotted that in actual fact, they form lanes. And you'll see this next time you're in a crowd, if you haven't spotted this already. You naturally fall into the slipstream of someone ahead because they buffer you from oncoming interaction. And this allows more smooth traffic through these environments. Similarly, we organize traffic in other ways, such as on highways, where there's more regimented control, such as driving on the left or the right and following lanes. So in addition to developing highways, we also develop, this is my favorite bridge, we also develop bridges. And so we've been able to modify our environment in a variety of ways to allow our society to function in a more efficient fashion. Yet, if you want to move this bridge, if, for example, it's no longer required at that location, but needs to be moved somewhere else, well, that's incredibly difficult. One of the great challenges with the railway system in the UK is the strong dependence on the history of the past, which makes it difficult to adapt to the present or the future. So are we the only organisms out there to have traffic infrastructure? Well, you might not be surprised in this audience to find out that we are not alone. And in actual fact, there are remarkable examples in the natural world. So one of my favorites are amia. These are near-blind individuals. In this case, it's Eceton hematum, and I also study Eceton burchellii in Central America. So these are near-blind individuals that are using both chemical communication, following the trails laid by others, but also the physical contact with each other to coordinate their behavior. And this is an example of a soldier caste within this species. It really is, as my PhD advisor Nigel Frank said, the blind leading the blind. Yet somehow, when you have millions of individuals or hundreds of thousands of individuals coordinating in this way, they can exhibit extraordinary collective behaviors. And so here's a picture of Nigel. And I just want to thank the two mentors that are centered around the two systems I'm going to talk about today, the army ants with Nigel, and later on, I'll discuss with the locusts. Because I'm typically, when I give a talk, I always thank my students, my postdocs, who are amazing. But today I want to thank my mentors, like how I got here. And so when I started working with Nigel, I was really fascinated by this behavior. And so I went to Panama and I filmed ants and I tracked their movements. So here we're just seeing one ant returning to the nest, here colored in red. And just to give you an indication, the nest is towards me. So it's going from the right to the left. And what happens is this ant happens to interact with 5 ants that are leaving the nest, which I've also tracked here. And you can see that when they interact with these ants leaving the nest, it really just pushes its way through. It's carrying food, it's burdened, it's like a truck, so to speak. And the ants leaving the nest are not encumbered, and they will turn sharply upon encountering this individual. Now, this is just an example of a few individuals interacting. What happens if we scale this up so we have many, many ants interacting with each other? Well, what we find is that the ants will spontaneously form traffic lanes. And unlike the human traffic lanes that we see that definitely allow us to have less disturbed traffic, the difference here is that there's an explicitly evolved behavior that we find to actually optimize the traffic flow. In humans, this would never happen. We tend to be selfish. We want to get home, and we don't necessarily care so much about the movements of others. But the ants have evolved to function collectively, and so they can exhibit this beautiful example of traffic organization. And so this level of efficiency was one of the first studies of how traffic organization functions outside us humans. And there have been many, many studies that have followed it since. But the ants don't just organize their traffic, they also do all sorts of other really cool things, like build bridges out of their own living bodies. If we look at army ants clinging together, you can see how strong these connections are. They've got evolved tarsal claws that interlock, allowing them to really span and stretch and contract and to do amazing things. They, in fact, build the entire nest from interconnected ants because they raid across the forest floor and then they have to move on. They have to be nomadic. And so they can't afford to dig a nest and build that infrastructure because they have to move on. So they do it in a living architecture. You can see here part of the nest, their bivouac. And so we studied how these bridges form by allowing the ants to form bridges in environments that we can control. For example, the distance between the gap size. We could also measure or even control the flow rate crossing the gap. And you see how resilient it is. They initially made a mistake. They now recover from that mistake. And they can expand across extraordinary gaps using simple local rules. And here you can see in a time-lapse movie, the bridge can actually move to become more efficient. It's a trade-off between how many ants you want to invest in your bridge versus how much shorter you want the path to be. But there's no individual ant. The queen is not controlling this. There's no individual telling them what to do. They are just evolved to use very simple local rules. And here you'll see another example of a failure in a moment. But don't worry, they will recover from it. You can see them carrying the brood over. There we go. But they'll get back. Yeah. And so when we record, how do the ants do this? I mentioned these local rules a moment ago. But what they use are their antennae to measure the flow over them. They're not counting explicitly. They're not, they're just getting a general sense of how dense is the flow above me. How often am I being hit on my antenna by other ants? And you can see that the time the ant chooses to spend in the bridge is this non-linear increasing function of traffic, which means under high traffic conditions, these bridges get large and very stable. But if the traffic goes down, the bridge will just simply dissolve and form elsewhere where it may be needed. And it's not just the traffic flow that's important, it's also the connectivity among the ants, how they connect with each other. If you're in the middle of the bridge, connected to many ants, you're much more likely to stay than if you're at the edge of the bridge. So this is how the bridge can move. The edges are very malleable. The edges can grow or recede, and this can allow the bridge to move. And if you happen to take that shorter path across the bridge, because it was shorter and your pheromone is being laid, it's more likely others will follow that shorter path and reinforce this over time. It's not instantaneous, but these bridges can move. And I'll show you the video again here, this time lapse of the bridge moving. And to give you an explicit sense of the time scale, it's around 20 to 30 minutes that the bridge will slowly move. And it relates to various factors, such as the angle of the zigzag structure. If the angle is very small, then the gap is less, and so the bridge can move more quickly. But it's not just about the geometry, it is also about the flow of the ants. They're more likely to move when they need to move. So there's a direct trade-off between investment in the bridge and allowing the colony more efficient movement. And so the colony as a whole, we can show in this paper, actually is provably optimal. But no ant knows that. They're just aware of the local interactions that they've evolved to function in this way. And we then observed something, this is the cool thing about science, you just see random things and it gives you an idea. So here we saw army ants raiding on an electrical box in Panama. And there's a colony of ants inside that box that they're attacking. So I might transition to the adapter from Apple in case this continues. And you can see here that there's a different structure here. It's not a bridge-like structure because it's on a vertical surface. But the ants, nonetheless, you can see some ants are static and other ants are moving. And so this gave us an idea. Perhaps the ants can also form scaffolding. Perhaps it relates to the slope in the environment and also potentially the traffic. So we developed a setup here. We could actually manipulate the slope to change the cost of ants falling, or the probability of ants falling. So if you imagine an ant encumbered by food returning to the nest on a relatively slippery surface like this, could readily fall. And so here you can see the type of structures that form here. And we could show that this, of course, prevents the ants from falling and losing their valuable food. They really are on an energetic tightrope, as Nigel Franks had previously shown. And so here, if you look at different timescales, you can see in the bottom here, the ants choosing to stop and support the other individuals. So together, the army ants are solving collectively remarkably difficult problems. There's no individual that's telling them what to do. Each individual is responding to local rules, and together they can solve a variety of different challenges. But this is because they're very closely related to one another through haplodiploidy. Now that's amazing. They can do such amazing things. But there are also other types of swarming insects that are genetically selfish, where there isn't selection operating at the level of the group. I mean, in the ant, it's also operating at the level of the individual, but you can think of it to a first approximation of being the group. But in other systems, it really is about the individual within the collective. And in the second part of my talk, I want to talk to you about another system, and these are desert locusts. So one of the biblical plagues, one of the plagues of Egypt. And you can see that these form vast swarms. As far as you can see, the individuals are moving apparently in unison, as if one mind. And I did my field research when I was based at Oxford in Mauritania. I'll show you some images of that. But this one species, the desert locust, can invade up to around 1/8 of the Earth's land surface and is estimated to impact the livelihood of 1 in 10 people on the planet through impacting food security. So it's a major problem, yet because it's impacting poor people, there's relatively little research going into this. And so I thought, you know, it's a public lecture. I thought there might be kids here, and so I didn't know what level to aim this at. So I do apologize because there's so many luminaries in entomology here. But some of you may not know, maybe one of you may not know, that you know, locusts are actually, you know, short-horned, cryptic, shy grasshoppers that actually don't really like to be together. If they can feed independently and be isolated from each other, they're just fine. A little bit like the grasshoppers that may be in your local environment here in the UK. However, they also have this ability to transition as they molt. They change their coloration, but behaviorally, they will change within a few hours to, instead of avoiding each other, being attracted towards each other. And as they molt, so these are juveniles, they will change their coloration and all sorts of physiology. And you can see this aposematic pattern, and they also will tend to eat more toxic plants. And so it's almost like, you know, it's remarkable. It's almost having two animals in the same genome. And it's extreme phenotypic plasticity, allowing the animal to switch from one type to the other. And this is an evolutionary adaptation to living within incredibly variable environments. And so imagine in Africa, there's monsoons coming in, bringing plenty of water. Well, this can change the landscape very quickly. It will green up over very large scales. And as this food availability changes, as you can imagine, the solitarius population can simply reproduce, be happy, and grow. So under such conditions, the solitarius individuals will grow in population size. And if there were any gregarious individuals, they would tend to transition into the solitarius type. And then across generations, they would remain solitarious. And so this can be a long timescale process of the solitarious population building up and building up in the environment. But what if the population of these insects gets so large that that food starts becoming a scarce resource? Or probably what's more likely in combination is that there can be sudden droughts in these environments. You cannot rely on the rains coming regularly. The rains can come and then be gone for a long time. So you now have a big population of solitarious insects and not enough food. This forces the insects to come together. This forces competition. This forces the animals to have to migrate to get out of nutrient-poor areas. And they transition into this beautiful gregarious moth. What an incredible animal. Look at those eyes. And these insects will start marching together, forming vast swarms, can extend over many, many square kilometers, hundreds of thousands or even millions of insects. It really is something to see. So they march together across the landscape. And what's also amazing, and this was discovered by Steve Simpson, who I'll show you in a moment, that mothers even put a chemical in the egg foam around the eggs to inform the eggs, so to speak, what environmental conditions to expect. So when they emerge, they can be ready for a gregarious or a solitarious lifestyle. So what is known about the mechanism that drives locust swarms? So when we first started studying this, there was really almost nothing known. There was an amazing researcher called Peggy Ellis, who'd done a bunch of work in the 50s and 60s, but it was published in the Anti-Locust Bulletin, a sort of obscure journal. And at the time, her work wasn't recognized for what she was trying to do. She was really the first person to try to think about things collectively. We all had locusts in our classrooms or in our college, our universities. Sorry, I've been in America too long. In our universities. And the locust is one of the best studied organisms, like Malcolm Burrows' work, for example, for individual neurobiology. But Peggy was the first person really to try to see what happens when you put them together. And if you think about the army ants I showed you before, if we had the entire of humanity trying to study a single ant, a single army ant, using all of the modern genetic, molecular, microscopic tools, it could never allow you to predict what happens at the level of the colony and the behaviors at the colony level. So this gives you an example of how you have to think in this collective way, looking at these organisms. And so, as I mentioned at the beginning, these are not cooperating. It looks like they're sort of cooperating. It looks like they're doing things together for the benefit of all, but that's not the case. And so before I go further, I want to thank Steve Simpson, who was my mentor when I did my first postdoc at Oxford, and he really got me excited about the locusts. And he's still just a very dear friend, and I'd just like to thank him for all of his amazing advice over the years. And so working with my first PhD student, Sepide Bazazi, we discovered that these so-called vegetarian insects would frequently exhibit aggressive interaction. So we'd be filming them, how they move, and they would frequently bite each other. And I thought I was crazy because the number of insects coming out of the arena at the end of the day was less than I put in the morning. I thought, maybe I can't count, it's quite possible. But in actual fact, what we found was that these insects were highly aggressive towards one another, especially when deprived of protein. And so here's the sort of damage that you would see. And so even a little bit of damage on the abdomen that you see here, this would really be a terrible thing in a desert environment. And of course, locust bee there is really in trouble. And so what we did was we did experiments where we could cut a little hole in the belly of the locust, and we could then cut the nerve that gives the locust sensation to the abdomen, so it can no longer feel the biting of others from behind. And we did control experiments where we located the nerves and we tickled them and we sealed it up again, but we didn't cut the nerve. So those insects can still feel the contact from behind. And then we could put swarms together of these two treatments. So we have the control in black and nerve cut shown here in red. Now, this operation didn't really seem to harm the locusts. They're quite resilient creatures. If they're in isolation, the way they moved, the way they fed, their behavior was really very, very similar. However, if we put them in groups to see how their group behavior was, you'd see a massive difference between the nerve cut individuals and those that could feel the biting behavior. And locusts will tend to march for around 8 hours a day. And here you can see locusts in a swarm, so many locusts in an environment. One swarm is nerve cut, the other swarm is controlled. And you can see here that the control insects that have this biting behavior, they activate each other and they march. However, the nerve cut individuals will become quiescent and will not form swarms. So by removing that biting behavior, we've removed this feedback loop that causes the swarms to start moving. But it's not just about contact. The visual system is also critical. So this is a super high-tech experiment where we painted parts of the eyes of the locusts. So this locust has got normal vision. These locusts have the front half of the eye painted just with model paint that you paint, little model planes or whatnot. This one has the back part of the eye blind, so it can't see those approaching from behind. And these poor locusts have the compound eyes completely occluded. And what we found was that the totally blind individuals, as you can see in orange, they couldn't swarm. But the back blind individuals, even though they can see those ahead, if they can't see those behind, this really impedes their ability to be able to form a swarm. However, that's not the whole story. We also found that the front was important. Seeing those receding from the front was also important. And we could measure the relative effects of these different parts of the visual field. So I decided, well, it's great to study the insects in this controlled laboratory environment, but ultimately, what I want to do is see them in the wild and study them in their natural environment. So there happens to be a big outbreak, a big plague in Mauritania. So I went out there and we did, thanks to our colleagues in the Anti-Locust Commission there, we found the locusts, and it was an incredible country to explore. Not easy, but an incredible country to explore. And here's one of the locusts on a tent. And that's me filming the locusts, and that's one of my colleagues, and that's our camel that we used to carry our equipment, an incredibly grumpy animal. And you can see in the background footprints in the sand. So the locust swarms can lay these footprints in the sand, and if there's no wind, you can actually track them for quite long distances. And this is the camel, because it's cool. But because there were, a huge outbreaks of locusts there, and we were very, very remote, and despite the fact that we had plenty of money, there was nothing to buy. There was no food to buy. And so we really got into a bit of trouble. And the only food we could get were camel entrails. This is not our camel. Our camel was not hurt. But you can see flies, on it down there. And we just hung it in a little tree to dry like a jerky. And I'd been a vegetarian for a decade prior to this trip. And actually, it didn't taste as bad as I thought. But my God, I got sick. I got very, very sick eating this. So you can also see how well prepared I was to compare my tent with my Mauritanian colleagues' tent. Mine cost like 40 quid from the local camping shop and had 10 pegs this big, which of course don't do anything in the sand. You have to drive them in. But nonetheless, what you're seeing in the background there is a sandstorm coming towards our campsite. This is the same tree we had hung the jerky, actually. This is a sandstorm coming. And the next morning, my tent was still up and the Mauritanians' tent had collapsed and they were all sleeping under it. But this lasted for about a day and a half. And this is not a sandstorm. This is exactly the same sandstorm that I found on NASA's website. And it literally blew the locusts away. I was there for 2 months. I got 20 minutes worth of data. I keep it in my office just to remind me never to do that again. It was It was a disaster. It was an experience of the best thing you could say about it. So again, with my colleague Steve Simpson, we decided to go to the United States of America, where they have roads and useful things like that. And we studied a comparable system, which are Mormon crickets. And so these are so-called Mormon crickets because when the Mormon settlers settled near the Great Salt Lake, things were going great for a long time. And then huge swarms of Mormon crickets came through, devastating their crops. And then God sent a flock of seagulls from the Great Salt Lake that chives down. And we actually saw this happening. We saw flocks of gulls coming in. And so if you go to the Mormon temple, outside is a beautiful stone sculpture of Mormon crickets with a seagull on top. And the seagull is still the state bird of Utah. And these, again, thought to be vegetarian insects, would frequently eat roadkill, for example. So they're chewing the ears off this bunny. It's normally around the length of their body. They're crawling in through the eyes and into the mouth. It's kind of gruesome, but also fascinating. And, we did not expect this. And they would also regularly chow down on their buddies. And in fact, this can lead to slicks on the roads there because they stop to eat the dead bodies, then they get run over, and then more insects stop to eat those. And it can actually lead to road closures with this slick of cricket entrails. And so we did an experiment where we manipulated the carbohydrate and protein composition of the food available to the insect. So O is a sort of zero nutrients. It's like the sort of McDonald's meal of crickets. And you'd think, these so-called vegetarian insects would go for carbohydrate, but they had no preference for it whatsoever. However, as the diet increased in concentration of protein, you see there's a much greater desire. And this makes sense because in these arid environments where they've run out of resources, they really are short of critical nutrients like protein, salt, and water. And when we analyzed the belly contents of the crickets in a swarm, we found only the ones really near the front, the first few meters, were the ones that had food from the environment. And it was largely proteinaceous parts of plants. So they're really limited. And this is a living histogram. Of course, I randomized it when I did the experiments, but I thought this would be cool to show in talks. And this is their preference for salt. They taste it with their feet, and you can see them fighting over 0.25 molar salt concentration, which is represented in the figure here. So what's special about that salt concentration? Well, that turns out to be the concentration of their blood. So they're finely tuned to a cannibalistic lifestyle. And so when you're in this environment and there's a high density of insects and there are no resources, especially these rare nutrients, what's better than a perfectly packaged other individual? It gives you everything you need, except perhaps parasites. So there's the strong selection again to have strong cannibalistic behaviors. So far from being cooperative, the crickets, and also we showed the locusts, are on a forced march. Stop, and you risk being cannibalized by those behind. So we were pretty happy about this. We thought, well, we've really cracked this problem. People didn't understand how the swarms were coordinated. Now we've got a really good understanding. And so then I moved to the US. And one thing is you cannot work on locusts in the US because if they escape from your lab, they could become the next biblical plague, and then you're in deep, deep trouble. So it was very difficult for me to continue the research on the locusts. So I started working on all sorts of other creatures like fish. Here are some sharks hunting in the Maldives. And actually, this photo is from one of more recent field trips, which was taken by Angela and August on our field trip that actually won the Royal Society Photography competition last year. So it also produces, I think swarms or collectives in general really attract the attention of the public. So it's a great way to communicate science. We also study wolves using, again, drone imaging and more recently AI technologies that allow us to track individuals in the wild, to identify them, to look at the social structure of these animals that are notoriously difficult to study using other means. We've also looked at the so-called solitary octopus to show, yes, they may be solitary with respect to other octopus, but they have highly complex social lives with a whole bunch of other species of fish that they almost always hunt with together. And despite this being a really cool story for lots of different reasons, the press just picked up on the fact that they also have a partner regulation mechanism whereby they will punch fish that inhibits the coordination of the group. So here you can see some punching. And Stephen Colbert particularly liked the fact that, you know, as a pugilist, having 8 arms might be a very helpful thing. And also, you know, we got to work in really very nice places. But then I moved back to Europe. In 2020, in January 2020, just before we were all worrying about COVID, like very fractionally before, well, some of us were worrying about it, but before the governments were doing anything about it, there happened to be a major plague in East Africa. This turned out to then be exacerbated by COVID because it meant it was very difficult to move people and equipment and chemicals around Africa. So we sent a team out to Kenya. We managed to get the second last plane back before the planes stopped. And so this is our little team that was out there trying to film the locusts. And we just used this as an opportunity to really see how does the swarm function in the wild. And because we have these new deep learning tools for tracking, like I showed with the sharks, we can now track all of the individuals in very large swarms under natural environmental conditions. And the work I'm going to show was just published last week. So I'll highlight this particular paper. And so the first thing we wanted to check was before we had this evidence that the biting behavior was really critical. But we didn't really know whether that was so important for the coordination. It definitely gets individuals on the move. But does it actually align the individuals? And so if you take a locust out of a swarm like this with the marching direction from left to right, as you can imagine, when you pop them back in, they will march with the swarm. If you remove their ability to smell, it doesn't change anything. In fact, we have strong evidence that they downregulate the olfactory cues. Things that they're normally super attracted to in the lab, put them down in the swarm, no response. Kind of amazing. Similarly, they have a very good polarized vision at the top of their eyes, which we do think is generally important, but for this type of coordination, we didn't find evidence for that. But if we blinded the individuals, they would march. This was the surprising thing. They would march really well in a very directed way, but just in completely random directions. So imagine you're in a crowd like that, you know, and you were to close your eyes. You could just simply feel the direction of the crowd. You could just go with the flow, but they don't do it. So just like they're turning off the olfaction, I think they're also turning off the tactile sensing. So vision is really, really important for regulating this collective motion. So it's necessary and sufficient. So how is vision used to coordinate collective motion? How do people think it is used? Well, there are many, many experiments. You can take pretty much any insect that at least I know of, and also most vertebrates, and if you show them moving dots or moving stripes, they will tend to follow them in what's called the optomotor reflex. This is innate, and it's extremely robust. And as I said, it's not just insects. Fish, for example, show this. And this is a paper from 2023 that says this innate optomotor reaction, the following reflex, ensures that fish maintain a single school during movements and rapid maneuvering. And in these studies of locusts, where moving dots are shown to the animal, it's just assumed that they must be using the optomotor response, and the locust, in actual fact, can't even turn. It's forced to be in alignment with these dots. So it's such a preconception because it's innate, it's robust, and if you look at swarms, you know, there's a huge amount of optical flow from the perspective of the focal animal. But no one had actually tested it. So that's what we set out to do. And another really cool thing, of course, is that we can rear them to be solitarious or gregarious. So if you're interested in group behavior or the evolution of grouping or just all sorts of aspects, to have the same animal that can be solitary and gregarious is a real gift. And if this motion following was an adaptation to being crowded and was an adaptation to being in swarms, we may expect that the gregarious insects would be more sensitive to such cues. So when we tested this in the classic optomotor response, where the animals will follow moving dots, we found there was absolutely no difference. If anything, the solitarius was slightly more sensitive. So this is the first line of evidence that following these moving dots may be different to the mechanism involved in coordinating social behavior. But those moving dots are not very realistic. How can we then explore the role of realistic motion in a highly controlled way? So what we did was develop immersive, volumetric, or holographic virtual reality for freely moving locusts. So we can embed locusts in a fully holographic swarm, really volumetric. It's hard to imagine. I'll try my best to explain, but it's like if you put glasses on and you're actually in virtual reality, that's how they see it. But we don't put glasses on, we put them in an environment that gives them this illusion. So it's called the anamorphic illusion. Now, you're super smart humans, yet this tape looks to you to have a volume above the table. It does not. I can even tell you it does not, but your brain still sees it in 3D. And there the illusion is broken. And when it goes roughly back into position, because the volumetric cues are there, your brain automatically thinks it's three-dimensional. So again, this shoe is not actually there. It's not 3D. So if we do this in a computer, we do this 100 times a second, and we track the eyes of the animal, we can have the animal interacting in a fully volumetric 3D environment of arbitrary complexity. And so this is the virtual environment with 3 projectors. The locust is on a big motion compensating ball, the biggest ball we could fit through the door of the university. And as it moves, the ball moves to keep it always on top, and we can immerse it in a virtual swarm. And so this just to emphasize, as the locust moves, we then rotate the ball, and so it always keeps the locust on top. It can march all day for its 8 hours that it wants, as it does in the field. And the ball is so big that even if it hops, we still track it in the hop in 3D, and when it lands, we just pop it back. So they're welcome to hop as well. And I can't really show you what the world looks like because I can't show you volumetric projection. I can't show you the fact that the locust can see all the way around, but this is the best I can do. Imagine this is fully 3D, like 3D models in front of you moving that you're interacting with. This is what the locusts are seeing and interacting with. So we could do the same experiment again, where we show them now optic flow cues that are not just arbitrary dots that people tend to show in experiments, but are actually the flow cues that they experience in the real world, but we've taken away, for example, tactile cues, so we can disassociate them. We're actually building a VR system now that can also tap the locust to give them both. And now we see a massive difference. The gregarious will march readily, but the solitarius no longer follow the stimuli. In another experiment that we've done, which we haven't published yet, if we make a swarm of solitarius individuals, neither solitarius or gregarious will march with them, even though in the optic flow stimulus, they respond incredibly strongly, so they can see the stimulus. So they really are processing the visual information in a volumetric way. The 3-dimensionality is very important to understand their natural behavior. So overall, how we really thought that locusts are coordinating motion was really wrong. And I find this exhilarating because this allows us to start again and see what's actually happening. And at around the same time, in the last sort of 5 years in particular, there's been an explosion of incredible research looking at the neurobiology of spatial navigation in both the invertebrates, specifically Drosophila and a few other species, and also recently in the vertebrate. And what they have is a ring of neurons within the brain that define the direction of travel that the animal will make. And this is work from Vivek Jariaman at Janelia from 2014, I think, this paper. And you can see this ellipsoid body. It's like a donut shape with this electrical activity in yellow. It actually indicates the direction and the strength of the vector. It's astonishing. And you know, when I was working on fish schooling and all these other models, I was doing all these vector calculations. And so I suddenly realized, oh my goodness, this is like a fish school. This is the same sort of principles mathematically as we see within animal groups. And this architecture is just absolutely mind-blowing. So we developed a theoretical model of how insects and vertebrates make decisions. So in this ring attractor, there's excitation among units that are close together, that have similar directional preferences, and then long-range inhibition. And so it's called the neural ring attractor network. So just to represent it schematically here, if the red is a firing bunch of neurons, it will tend to excite those around it, but tend to extinguish the others, to sort of fight with them for control of the direction of travel. And what we could prove mathematically is that this ring attractor will always choose a single direction, which is a great thing. You don't want to be stuck in your brain with two options, because then you're going to get stuck between them and not know what to do. And in the absence of external cues, it's completely random which direction will be chosen. But if there are external cues, they can impact this ring attractor by causing electrical activity in the respective area of the ring. So if you imagine an individual moving towards two targets, up those X's at the top there, what you're seeing is the perceptual layer. So that's, in this case, visual information. It could also be olfactory, it could also be acoustic. But that input goes into the ring in the respective position of those targets with respect to the animal. So it's an egocentric perspective. And then these neurons are fighting with each other, competing with each other to determine the direction, the consensus where to go. And below a critical angle, they will come to an average consensus, but above a critical angle, one of the two subsets will tend to win out. Just that I'd found much earlier looking at animal groups. And so we could then design an experiment in virtual reality that would be impossible, of course, in the real world, where if a locust has a swarm moving on its left and on its right, then all of the classic models, the cousin model, all of the classic models will predict they will go with the flow. But the cognitive model doesn't. It predicts that they go orthogonal and then they swarm when they're within the group. So we now have an experiment where we can literally test the two hypotheses orthogonal to one another. So just to remind you, the locust is marching in its motion compensating ball, immersed in its virtual swarm. And this is kind of what it sees. Again, I can't really represent the volumetric aspect, but the models, classic models will predict it goes with the flow, but the ring attractor model doesn't. It predicts you should go one way or the other. And so this is the theoretical prediction from the ring attractor, the cognitive model. And those are the data. And of course, we did statistics and so forth. And we also then did experiments with large numbers of locusts in our imaging hangar, which I'll show you shortly, where we can track up to 10,000 individuals within swarms. And we showed that this is the mechanism by which they coordinate their motion. And so classic models, like the models I developed back early in my career, really are fundamentally wrong. They do not capture the correct behaviors of the animals. And it was really nice that Steve Simpson, having been my mentor early in my career, then wrote this beautiful piece to accompany our paper last week. And so, as I mentioned, we can now track up to 10,000 individuals. And now you're looking at time lapse. This is the first time anyone in a lab condition has been able to get the swarms actually forming, these mobile bands actually forming. And this is a bit fast, but this is just showing using deep learning, we can track all of the individuals. We can also come up with predictions that if there are two targets, those black stripes there, that the cognitive model predicts that the locust should collectively oscillate between the two options. And the nice thing about having a quantitative model is we can explicitly just test these predictions. Again, classical models of collective behavior would not make this prediction. And Tom Scott, a British YouTuber, came to the lab, and within 2 weeks, almost 3 million people had seen us running these experiments. Like, I mean 2 weeks after we actually did the experiment. So we want to reveal what we're doing kind of in real time and make the data available rather than hiding what we're doing until this very laborious process of publishing. And we're also going into the brain now to understand really what's going on. And finally, we're also tracking individuals in the field. How on earth can we do that? Well, working with colleagues in Israel, we've developed tiny, tiny tags. So these bats that we tracked in this paper are about half of the size of your thumb. And we can track them over 15, 20 square kilometers across their entire range in real time. We get the data in real time, so we don't have to recover the tags. So it's perfect for tracking these little locusts. And so all of this is happening in Konstanz. It's very close to Zurich Airport, about 45 minutes from Zurich Airport. So if you're close, do come and visit us. It was not bombed during the war because it's on the border of Switzerland, and they literally left the lights on to fool the Allies. And so it's a beautiful old city. And we have this very cool interdisciplinary center studying collective behavior from neural collectives to human collectives, physical collectives. And also a new building. It's incredible that Germany actually funds buildings for organismal biology, in my opinion, at least. And so we have this wonderful building with this 15 by 15 by 8 meter imaging hangar that we've tracked bees, moths. You can even track a Drosophila in this space at sub-millimeter precision, 100 hertz. So it's a really unique facility. And like all of our technologies, we're making it available to the whole community. So you can simply book it online, come to Konstanz, and we will help you set it up. We have a full-time technician. We'll provide all the resources to do studies. I think it's going to be brilliant for ants. We're starting a whole bunch of ant projects this year. And this just gives you a sense of the scale. That's Tom Scott when he was visiting. So you can really start studying animals over the range that matters for them, including animals like birds, which are obviously less important than insects. But the cool thing is, these technologies, as I showed with the sharks, can now be applied in the wild. So these are the sorts of birds you find in the garden. These are great tits, looking at how they see each other, how they observe each other, how they copy the behavior of other individuals. We can reconstruct their retinal fields of view. And coming back to the sharks, we can not just track the sharks, we can also even get estimates of the flow of the animals within these groups, which is really remarkable. So an incredibly exciting time to study biology in general, collective behavior in particular, and insects especially. And thanks very much for listening.
Yeah, that's mind-blowing, literally astonishing. I'm sure we've got lots of questions. Who'd like to think more?
Thanks very much for that amazing talk. Near the beginning, you had a very beautiful three-dimensional graph of the tracking efficiency of the ladies.
Yeah, of the army ants, yeah.
Yeah, I was wondering if you could possibly explain a bit more about how you quantified efficiency as a variable.
So the efficiency here is just the rate at which you can transport ants both back to the nest and out to the raid front. So it's really the same as traffic efficiency. How much flow can there be? You could, of course, prioritize as the ants do returning traffic, because that's the traffic that's carrying food. But overall, it's really the overall efficiency. Whereas in humans, of course, wouldn't it be wonderful if on highways we use strategies that cared about others' efficiency? We just tend to get there as fast as we want, the ants do a much better job. Yeah, I guess. Well, I'll go from the front and then the two at the back.
You mentioned between the locust, the solid phase and the gregarious. You said they were attracted to one another when they become gregarious. What was the attraction initially once they become gregarious? I know the environmental pressures to turn into, but how do they then get attracted to each other?
So in terms of the brain, like how the animal goes from sort of repulsion to attraction, you mean? Yeah. It's a brilliant question, and I think there's a clue there from Drosophila. So recently it's been discovered that Drosophila actually have a switch to turn attraction to repulsion or vice versa. Now I'm not saying that that's what's happening in the locust, but at least it's a clue that you can actually have a very, very simple mechanism where you just change the sign. So for example, this is really cool. So if you want to work out where you are in the world, or you want to do path integration, you're an ant, you're going around, and you want to then, when you find something, go straight back to the nest. You need to keep a record of what you're doing. You need to add up all those vectors. You need to do vector arithmetic. Then you need to invert it, and that can give you the direct path back to the nest. right? And it turns out that's what they do. So they have there's two ring attractors. So they take the information from the ring I showed you, they put it into a cosine form in another ring. And if you add a cosine to a cosine, you always get a cosine. So basically, just by adding these together, they're doing vector arithmetic. And then all they need to do is simply flip that, and then you can go straight back. So they find these beautiful solutions for sort of solving complex arithmetic problems. It's called phasor arithmetic in this example. And we think locusts are probably doing something similar. However, and I was going to have this slide in, we've just got 2-photon imaging working for locusts. It's been very difficult. So also Bill Hansen's group in Jena has managed to get this working for a different species of locust. So now these non-model organisms are becoming more tractable. So if we can get more funding, we're really going to go into the central complex and really start asking whether it's the similar mechanism that's driving this. But I don't think it needs to be complicated. You know, and effectively, in summary, collective motion, you know, all of these models I came up with early in my career where I guessed, you know, this repulsion alignment and no, it's all nonsense. It's actually already there in the spatial navigation circuit. A tiny little tweak and you get collective behavior. And so I think there's going to be little tweaks that the brain can do to turn these things on and off. But there's going to be all sorts of other cool stuff happening. In fact, there's some experts in the room here who work on this, this transition that can probably tell you much more than me. I think there were two questions at the back. We'll start on the left.
With the Mormon crickets, you said only the ones in the first few meters of the swarm have really had much of the rare nutrients.
Yeah.
Do they show any behavior when they try to skip to the front of the queue, or is that too energetically costly for them?
Yeah, so it's a great question. And in actual fact, if I were to show you that video again, if it's not going to black out, what you would actually see are most of the insects marching this way. You don't see this in locusts ever, but in Mormon crickets you do. Most of them marching this way, but some of them marching this way. And so why are they going counter? And they've got heavily armored heads. And they just go straight through the swarm to the back, and then they molt. Because it's really dangerous to molt at the front, because you're just going to be eaten. So there is a kind of a rolling swarm ongoing. And as individuals are eating, they can be overtaken by others. So there are different aspects. So they are, in a sense, trying to get to the front. But when the swarm is, say, 5 kilometers long, and it's only the front few meters that's getting any food, it's probable that many of them are surviving through cannibalism for long periods of time at least.
Curious about the locust virtual reality. What was the process of developing that and applying it to the technology you had in the lab? And do you see any applications for the engine or the programming you made elsewhere?
Well, lots of failure, I would say. So I tried this a lot when I was at Princeton, but I didn't have the resources really. So when I went to Max Planck, I didn't give up and we really pushed it. So in actual fact, in our work, we've developed virtual reality for locusts, but also for fish that can freely swim. And so it's fully 3D volumetric. We developed it for flies. We're doing a lot of work now on ants. I don't know why I worked on flies. Ants are so much better in virtual reality. So we're getting, I mean, the answer, the data is just so clean. So yeah, so we just love developing these types of things. We're also developing the world's first 6-axis of virtual reality. So for hawk moths and for bees that can fly quite strongly. So we can actually allow the animal. So our virtual reality for flies, the animal is magnetically tethered and can just rotate. That's rubbish for a flying insect. They really need to have the degrees of freedom. And so now we can begin to actually have flying locusts, flying moths, hawk moths, hummingbird hawk moths in virtual reality too. So, and applications, I don't know. All of our stuff is free, open source. We put all of the design is basically 3D printing and very simple stuff. So that virtual reality system is 33,000 euros. Our new virtual reality system is less than 100 euros. So we're also really focusing on making it cheap and accessible. And we're currently testing it in India. We've developed kits that we can send around India. So people, because if you think of a school kid or college student, university student, you've got biology there, you've got a bit of computer science, you've got engineering, you've got physics, you're building something, you're understanding something that no one else has studied before. Most insects have never been studied. And so I think it's a really exciting way to also educate. So this is one of our programs.
Nice.
You're welcome.
Hi, and yeah, good talk. Good to see you again.
Yeah.
One of the things I found really interesting was obviously the visual cues are really important for those gregarious locusts.
Yeah.
And they're very strongly patterned, aren't they? So do the locusts
recognise that happening, and can you investigate that by painting the locust or doing something?
So we did something similar to that. So you're absolutely right. So we can map any mapping of patterning onto the body of the locust. And I sort of stupidly, naively thought it wouldn't really matter. Like if the locusts are this gray or the average color of those locusts, it will be just fine. It's not, right? So for example, if we make them solitarious looking, even though they can strongly respond to that contrast difference, as we can prove in optomotor stimuli, they do not swarm with them, right? And it's not just that. Like I spent a long time in Oxford back in the day making two-dimensional images of locusts marching and trying to put a locust in the middle and letting it go to see what it would do, and they didn't do anything. But as soon as it's in 3D, same with the fish, by the way, as soon as it's in 3D, bang, you get the strong response. And so, really, the patterning is, I mean, we haven't published this yet, so I need to be cautious, but I think it's almost certainly not just aposematic. Like, I never, ever saw predation in the field. My colleagues that went out in 2020 never saw predation in the field. I know Greg sort of seen it, and you know, it might be in certain environments, but I don't think the aposematism is the only story. And so Greg is working with us on this as well. So yeah, that's another paper hopefully coming out this year.
Yeah, I'm just interested whether there's an applied angle to this. It must have been asked in the past. You know, is there something, the components within this which might provide some crop protection in terms of swarm disruption, even on a small scale?
Well, I mean, my focus is typically on sort of really blue skies work, but of course, working on locusts, one hopes that perhaps it might actually be, you know, it might also, in addition, have some utility. And you know, for example, the FEO have been using the wrong type of models. We've been using the wrong type of models to predict where swarms will move. But you know, I think to really crack this problem, we need, you know, we need botanists. We need physiologists, we need behavioral scientists, we need satellite imagery experts, we need climate scientists, we need weather. You know, I think it's such a big problem. And to really solve it, I think we need this holistic approach. And I just think there's no will because there's no money in it, really, ultimately. So I don't think we can do it alone. I really try to encourage, you know, as many people to sort of get interested in this problem as possible. But I've kind of lost hope over the years that we can really put together the team that would be needed to make a real stab at it. Because I think it's really in the prediction of where the swarms will go that, you know, but also potentially in the neuroscience, you know, who knows in the future, it might be possible that there's something that you can have a more targeted approach. But my feeling is that honestly, if you could find the animals, that's the really challenging thing. You know, there's many ways to kill them, but finding them is really hard over the scales that, you know, and also they're often in areas that are like Yemen and other places where it's just inherently really difficult to, you know, have control efforts be effective. So I think we're doing a tiny little piece of the puzzle, and it would be great if there was a more coherent effort put in.
You tried looking at swarm behaviour in like the African army ones, but Dr. They've got a solitary form and a gregarious form.
Yes, yes.
There was experiments tickling them and they didn't find that triggered.
Yeah, and there's other army ones as well. So so Greg is beginning to work on them because there is a pest angle there, and Texas A&M ideas there's a strong incentive to do stuff that's applied to maybe, but there's not been much on that.
Yeah.
But certainly there's, I mean, it's a very interesting question also with other locust species, whether, I mean, like the red-legged locust in the US used to form massive swarms. It's still extant, but it doesn't form swarms anymore. And that's probably because of the fragmentation of habitat. And so we're now doing experiments with Circuit americana, for example, that's related, but, you know, and does it really have no phase change or maybe a little bit? You know, so these are studies ongoing, but you're right, there's so many other wonderful systems that may exhibit extreme phenotypic plasticity that would be fascinating to look into. Oh, I'm struggling here. Maybe the gentleman at the back, and then I'll come back around to the front.
It's a planet that was moving in 3 dimensions with apparently no direction cue. Let's say like a Starling operation.
So I didn't catch the start of your question. I'm sorry. I didn't catch the start of your question. Please repeat.
Apply to a system that's moving in 3 dimensions with apparently no direction cue, like a Starling murmuration.
Yeah, so I think I think, and this is again speculation, I think it will apply. The model works in 3 dimensions very, very easily. And so the model can be turned from a circular model into a torus, into a toroidal shape. And then it can deal with 3-dimensional information pretty easily. The one study that we've done on 3-dimensional systems is zebrafish, juvenile zebrafish, where we do a lot of work looking at the brain and looking at schooling. But they do everything in 2D. It's quasi-planar. They'll choose a random depth, and they'll school always in a plane. And to disassociate socially, they disassociate up and down. And so if you think of the starlings, when you first look at a starling murmuration, it looks very 3-dimensional.
Yeah.
But my colleagues in Rome, Cavagna and colleagues, have imaged that in 3D and shown it's not. It's a flying sheet. So it's an illusion. You know, just like if you take a comb and you move it, you see this wave crossing, like Moir effect, I think it's called. That's the same effect you're seeing with the starling. So starling flocks are not actually 3D; they're almost 3D. But unlike the zebrafish that school like this, they flock like this. So it may well. So there's really not much known about this yet. Okay, down to the front.
I wanted to ask, how do you rear, like, what's the process for rearing the gregarious and the solitarius?
So the question, just if you can hear, was like, what's the process of rearing the solitarius and gregarious? The solitarius is a pain. It requires a lot of work. And the gregarious, we discovered that there's a huge warehouse near Munich where they breed locusts en masse for the food industry. And the quality of the animals is phenomenal. It's much better than when we try to rear them in large numbers ourselves. So when we work with large swarms, we buy them in. All the solitarius, we carefully look after every little one.
You were talking about following dots not being enough in the...
Yeah.
When you then developed the virtual reality, did you jump straight to 3D models of the same species? Or did you, like, explore any middle ground? Would they follow 3D sticks almost, or in between?
So you know, you think like the zebrafish being a vertebrate might be more sophisticated, but the zebrafish will follow any damn thing as long as it's roughly, it doesn't even need to be volumetric, as long as it's in 3D. That's really important. You can have a disk, a flat disk, but the zebrafish uses the Z-plane to get 3-dimensional information. If it doesn't have that, it's really in trouble. With the locusts, however, they will not respond to random, say, spheres. We started off with spheres and things like that, and they will not respond to it.
What about other types of invertebrate? Anything close, more similarly related to locusts themselves?
I don't know. It takes so long to do these experiments. We haven't exhausted every possibility, but we've looked at things like changing the coloration. We've also looked at changing the movement of the legs. You know, are they picking up on that frequency of movement, or is it, you know, things like that we've looked at, but so that's because we had one very expensive virtual reality system. Now we have these much smaller, much cheaper systems. We're building 100 of them. So now we can have 100 experiments running at the same time, so it can be much higher throughput. Plus, locusts can be interacting with other locusts in real time in the virtual environment. So they can be interacting with virtual avatars of conspecifics in real time. So this is something we've done very successfully with fish, and we're now trying to do with locusts. So once we have higher throughput and cheaper devices, then I think we can answer more questions. But you're right, there's tons more to ask.
I'm wondering how small you can go with the systems in terms of insects, you know,
Well, fruit flies are pretty small.
Okay, so you can.
I mean, fruit flies are small enough to really make PhD students never want to work in fruit flies again. It's you know, and I think, you know, and also, you know, some ants that we worked on, like Leptodorus acerborum and so forth, are pretty small. So I don't know how far you could really push it, because there is a physics for the stabilization of the ball. So for the small insects, we actually use it floating on air rather than moving it. But it's never completely friction-free, right? The animal needs to still exert some force for it to be able to work. So there will be a limiting factor at some point. But so far it's worked for sort of little creatures like flies quite well.
Around about the middle part where you were talking about the ants, you suggested an assumption that they had this cooperative behavior due to the fact they are more connected to their sisters than they are to their children and offspring, which led me to believe that you'd make a comparison to there being a cannibalistic approach mattering more about offspring than sisters? Is there any kind of like connection you put?
Yeah, there is a connection there. So the selection here for these locusts is really just out for yourself. You just want to get there, you want to breed, you want to have offspring. You do not care too much about other individuals. There's not sort of other regarding behaviors. Whereas the ants have evolved to actually exhibit these behaviors, like being able to build bridges and optimize traffic flow, because they don't have that sort of evolutionary constraint, so to speak. That doing things together as a group benefits the genes going forwards for the whole colony. Whereas that's not the case for the locust. It's each one out for their own to a first approximation.
How does sex tie into this? In the sense that, do they reach physical maturity? Do they reach out whilst they're swarming, or are they delayed? And can you investigate mate choice?
So it's really hard to look at the adults because the adults fly. And so there is no study. Well, there's studies by the British Army back in when they were in Africa, which are actually the best studies even to date, where they sort of describe the direction that locusts are flying within the swarms. There are imaging tools that I think could be applied, but it's so difficult to get permission to get technology into these countries. And you have to be really opportunistic because you don't know where it's going to happen. And even with the big imaging hangar, I don't think we can do the flying swarms. So flying swarms, I think, is a super interesting thing. Someone should do it. Hopefully, it will be us at some point. But the mating, our colleagues, Abdallah Baba from Mauritania, who was also with me in the field, had a recent paper 2 years ago, I think, in PNAS, where they showed that desert locusts have a lecking system in the wild. And so what we did, again, with my colleague Greg Sword, in this big environment, we set up to try to replicate as best we can a lecking setup. And I would say we were marginally successful. So we're going to try again. But certainly they compete for positions on perches, and then there seems to be selection among the habitat. And that's kind of cool. Yeah, we're on it. We're trying to study that aspect as well. But in the field, it's really hard. I mean, I saw them mating in the field, but it's really hard to be able to predict where they're going to go. And the egg-laying fields, in many cases, are unknown. So the control efforts are so poor because it's hard to predict, because the flying swarms can travel in the trade winds and go really, really far. I mean, this is why in the Bible, they all flew out to sea, because that's what they do when they run out of land. They avoid polarized light generally, but if they run out of land, they'll fly to sea. And they landed on the Beagle, and they'll land on ships, so forth, just trying to get to new areas. But it's really hard to really figure out where they're going to land to make.
Just take one last question.
Okay, one last question. So you can fight amongst yourselves. Or come and chat to me afterwards, and I can answer any question.
Watching your locust, and they're obviously moving from an area of food exhaustion to an area of potential food, and that's the Mormon crickets are. What I don't understand is what is the motivation for following each other? I can understand now that when they're in a swarm, they are it's good to stay ahead and keep up with and not get eaten. But what is the selective advantage of all going in the same direction, given the nature of the distribution of food in the environment?
So great question. So we addressed this using an evolutionary model. So I didn't have time to talk about the evolutionary work that we did. But what we did is we could take the model of interactions, and we could then have, in our case, 16,000 individuals that are living in this virtual environment. And what we could find is that below a critical density, the evolutionary stable strategy was to evolve to avoid other individuals and effectively to be solitary. However, above a critical density of individuals, there were two things that happened. Firstly, they would then evolve to do a phase transition, or change morph into the gregarious type. And they would also evolve to come back into the solitarious type with a hysteresis loop. That's exactly what's found in reality. And so above that critical density, this is counterintuitive, right? Because in actual fact, marching together is the way you minimize cannibalism. If you imagine you're a car on a highway and you're going in the same direction as another car, you can be driving at 100 kilometers an hour and just completely fine. You start slowly adjusting your brakes and so on. But if you imagine the cars were going in all different directions and you were having to avoid them, especially if they were going to come and try and eat you, you'd really be in trouble. So it seems counterintuitive, but actually coming together and aligning not only gives you a chance of getting out of nutrient-poor areas, but it actually minimizes you're cannibalistic. So it maximizes your chances to cannibalize and minimalizes your chances of being cannibalized. And that's what selection operates on, the individual selfish strategy.
I'm afraid we are going to be kicked out of here very, very soon. So please join me in thanking Iain.